I've been studying aging really, well, I've been interested in it since I was 4 years old and set a goal in my life to leave the world a different place if I could, bend the needle towards improved health. And it turns out if we stay healthy and we're happy, we tend not to die. And so what I've been trying to do is to, first of all, convince the world that aging is something that we can understand and tackle, figure out how to measure it, figure out how to slow it down, and as I'll show you, we now have the ability to literally reverse this in the lab and even in people. So it's a super exciting time, and I'll share with you some of the entrepreneurial work that I've been doing for the last 20 or so years, trying to not just make discoveries, but make practical use of them as well. I just don't want all of us to have born one or two generations too early, because what's coming is incredibly exciting. And I think will increase lifespans by decades. So this is an equation that Claude Shannon came up with in the 1940s at MIT. This is what I think describes aging. And I get a lot of chuckles when scientists see this, because aging is thought to be something so complicated that you could never put it into an equation. But this essentially describes, or literally describes, the theory of noise being introduced into communication systems. And I have proposed and written about my hypothesis, which is called the information theory of aging, which is that we lose information. And that's essentially why we get old. But importantly, as I'll show you, I think there is a backup copy to the original information that we can restore and reset the age of the body and get our function back. So the analogy that I like, it's a little bit dated, but I don't have a better one yet, is a compact disc where the original music of our lives that's laid down as young embryos and children eventually becomes scratched. So you can see this music would jump around. And this is an analogy to each of the cells in our bodies, that we lose this ability to read the original information that we get from our parents. And this, I believe, is what causes aging. And aging, I've argued, is a disease. It's treatable. I think we all have family histories of this condition. But importantly, because we can now, I think, not just slow it down, but treat it, we should treat it a lot like, or regard it like cancer in the 1970s, where we discovered we could actually do something about it. Just because it's common doesn't mean that we should accept it, or just because it's natural doesn't mean that we should allow it to happen. We didn't do that for infections, cancer, or heart disease. So what type of information am I talking about? There are two main types of information in our bodies. The one that you would have probably guessed is the DNA, the genome, the blue strand there. But DNA isn't just a string of 6 foot of chemical in each cell. It's actually wrapped up in bundles by these proteins here, and they form chromosomes. And the way they're bundled or opened up determines the type of cell that cell will be and will remain. And you certainly don't want your brain turning into a liver anytime soon. And that's what this second type of information is called, or does. It's called the epigenome. And the epigenome is different than the genome in that it's largely analog information. It's three-dimensional structure over 4 dimensions over time, and it's vibrating very rapidly in the cell. The problem with analog information, for anyone who's ever lived in the analog age, tried to copy a cassette tape or Xerox something many times, photocopy it, you know that it degrades and you have a loss of information. And so what I'm saying is that I think a primary cause of aging, perhaps the primary cause, is the loss of this structure right here, the three-dimensional structure of the genome. And from all that comes the diseases that we're familiar with and many of the other hallmarks of aging that you hear about in cells. And one way to convey that is, this is a nucleus, the black strands of DNA. The purple and blue blobs are those proteins that package the DNA and tell a cell which genes to silence and which genes to turn on, as seen in these red regions. And you can see during aging, we can very accurately map the changes that happen. We think one of the drivers of this process are broken chromosomes. And so we can actually create those broken chromosomes and test whether that accelerates aging. So we generated a mouse. This is a mouse that didn't have its CD scratched, and it's now about 18 months of age, and it's a healthy looking mouse for its age. That's the untreated. What about the one where we cut the chromosomes for a few weeks when it's young and let the chromosomes heal? Do we get changes in the loops and bundles of the DNA? Do we mess up the epigenome? And the answer is yeah, we can create these scratches and we can make a mouse become about 50% older. And we can dial this up. We now have such control over aging, we can make it go quickly, we can make it go slowly. But the question is, can you really reverse it? And is there a backup copy of this epigenetic information? Can you get these bundles and loops to go back to the way they were? And the answer is yes, you can. We just published this in Nature in December. This is one experiment where the nerve in a mouse is damaged and the eye is on the right and the brain is on the left. And anyone who knows anything knows that the spinal cord and the brain and the optic nerve don't regenerate in adults, though in some species they do. So what we did was we put in 3 embryonic genes into these nerves, just a very painless and simple injection into the eyeball. We infect those nerves with our genes, the 3 genes, which we call O, S, and K for short. And we find that they are able to reverse the age and prevent rapid aging of cells that are damaged. And what we see is if we leave these longer, we can actually regrow nerves that are crushed. So we're looking at, in collaboration with Zhigang He's lab, the ability to regenerate spinal injury as well as optic nerve injury. But what's important is that we could also use this gene therapy in old mice, and we published that we could treat glaucoma and restore vision, and which is one of the leading causes of blindness, of course. We also put this system into old mice, just regular blind old mice, and we fully restored their vision as well, on average. So we also know that some of the mechanisms behind this and are developing other ways to reset the age of these cells. We think that it's a general mechanism because we've been able to, my colleagues and I, and increasingly labs around the world have been able to reset the age of various tissues and organs. This is just an example of what we can do. But how do we really know if we can reverse aging? How can I tell you that those eyes were younger, not just worked again? So we can now measure what's called biological age by measuring the chemical changes on the DNA that control those loops and bundles. These are essentially representative scratches on our CDs in our cells. And I could take your blood or take your skin swab and tell you, each of you, with pretty high accuracy, how old you are biologically and when you're likely to die. Now that sounds scary, but the good news is we actually can change this trajectory. As I mentioned, we have ways of slowing this down, we believe, as well as reversing it. This isn't cheap right now. A test costs many hundreds of dollars, but we've been working in my lab on this to democratize this test because we think if you can have your biological credit score, it doesn't so much matter what else you measure. This one number can help you with your life's trajectory. And so, first of all, what I think is coming is that we can one day turn back the entire clock in the body, but we're currently working, as I'll show you later, on reversing vision loss in humans. We hope that within 18 months to 2 years, we'll treat our first patient. So I talked about this reprogramming work while we wrote about it in Nature. So that's, if you want to check it out, if you're a scientist, it's up online. And then I also wrote about why I care about aging and why it matters and how I think aging occurs, what the future looks like, and why it's going to be important for the planet. And that's all in the Lifespan book on the right. And I get asked every day, David, how do you try to stay young? You don't look that old. What's going on? That's in Lifespan. So if you want to look at what I do on a daily basis, you can check it out. I'm working on pharmaceuticals as well. Life Biosciences is a company that has 3 programs that work on 3 different aspects of aging. One, I hope, is going to be a solution to obesity as well as longevity. There's one that works on proteins that get old and reversing that, destroying them. and that will be useful for aging, but also Alzheimer's disease and other age-related issues with the eye. And this is also the company that is developing the vision restoration. Metro Biotech is developing NAD-based pharmaceuticals. There's a lot of talk about NAD on the web. You're welcome to check that out. And we've been in human clinical trials for the last 2 years with promising results that I hope to talk about later. How do you measure the epigenome structure? So fortunately, there have been some breakthroughs, and this is a company that I'm on the board of and have grown since the beginning. This supplies kits to researchers and hospitals around the world to be able to measure your genome, your epigenome in 3 dimensions. It's called Dovetail Genomics. And I also, unfortunately, well, I don't know if it's unfortunate, but I did talk about the pandemic coming in lifespan, and this was back in 2019. And it happened, of course. But what it did was because I knew it was coming at some point, I started developing a company that can measure infections. Partly it was, if you read the book or have read the book, you know that our daughter, Natalie, our middle daughter, developed caught Lyme disease. And the test for Lyme disease is pretty useless and slow. Well, it's not useless, but it's definitely slow and sometimes fails. So what I decided to do is start a company out of my lab. It's called ArcBio. We built our own computers in my garage, actually, to run this. And now we can take any sample of your body and tell you what's non-human. And this is, I believe, is the future of microbiology and disease detection. Right now, this company is using their technology to look at viruses in the human body, and particularly in transplant patients where it's a problem. And this is my lab. I work with hundreds of people, but these are like my family. These are my students and postdocs who innovate and come up with these amazing things that I can then build companies around. And I wanted to end by thanking them, and thank you for inviting me to this wonderful conference today.